mechanism and how actually the molecules are relevant for some purposes. And these are some projects which were done by my students in Germany. Some of them are related to a computational approaches, like approaches to analyze adsorption of bacterial peptides on hydroxyapatite. So we also have one of the project here related to hydroxyapatite. Some are purely biochemical, let's say. Some projects are still in progress, like VCG protein. We are trying to model here, which is based on the experimental data. And some, which is particularly, as I see it, particularly important to, particularly interesting to do the robotics, maybe it's in vitro toxicity and mutagenicity of different substances, not related to in silico nanoparticles, but of course, graphene and in silico nanoparticles can be used too. And there are some slides from my previous publications where we have some big data, either this big data from the systems biology or the big data from the molecular, like structural biology, like the protein and how this protein interacts with various ligands. And so this is the analysis of the structural data. As you can see, the molecules, they are clustered. And we have two clusters, actually. One cluster consists of ligands which are good binders, and the other cluster consists of the ligands which are not really able to bind the protein, or they're not binding because the affinity is not good enough to reach the good binding at this point. And here's the analysis of the proteome. This is the human proteome, and for this particular research, I was trying to analyze the HIV patients and how these HIV patients are responding to some particular drug, and which genes are upregulated or downregulated, and how these genes are actually related with each other. Okay, so let's just summarize the two concepts. What we have here, we have on one hand, this is the some kind of a chemoinformatics paradigm. On the other is the bioinformatics paradigm, and what is the difference between these two? And sometimes it's really hard to discriminate which is which, because in both cases we're sometimes dealing with big molecules like proteins, which are the key point. For instance, if we analyzing the different substances, we would like to understand how the substances would interact with some particular targets in the body in order to treat some diseases like cancer, for instance, or the neurodegenerative diseases like Alzheimer's. And for this, we're using some proteins, and these proteins, of course, are known as a structure-based design. And if you are trying to analyze the ligands, then you can go for instance, for the ligand-based design. There are some other techniques as well, like a fragmentation of different substances. Like here, this is the algorithm. It's called Fragment Plus, which is dealing with the different substructures in a database. And of course, you can create something like a hybrid techniques, like combining different aspects of biomedical information, like chemogenomic database, when you're enriching the chemoinformatics data with the genomics data. And on the other hand, if you are dealing with the bioinformatics, you are usually dealing with some amino acids, genomes, genetics, genomics, and omics stuff, which is probably a bit different from chemoinformatics, as you can see here. Also in the realm of a big data, and for this you especially need robotics, because you have a lot of molecules, like sometimes millions and billions of molecules. So when you screening these molecules, you will come up with probably a couple of thousands or couple of hundreds to be suitable for the experiments. And logically to dealing with these molecules will impose some kind of a challenge for the scientist. So as it's shown here, we have a lot of like interactomes. These interactomes are actually from the publications we have previously published in a high impact factor, trying to investigate the molecular mechanism of some cardiovascular diseases like Takotsubo syndrome, such as here. And so we are trying to investigate how microRNAs are important to treat, trying to find a treatment against this disease. And this is just a spectrum, and on this graph it's just a spectrum of chemoinformatics and what are the key points, milestones of this spectrum. The milestones are information, knowledge, rules, number of facts and results. Sometimes they are overlapping with each other and actually describing the chemical data and collection data. You would try to analyze all this data by means of rules and the number of facts, which include molecular similarity, different number of rules such as Lipinski's rule, Farmakophore rules, and stuff like that. The number of facts also are including different algorithms like k-nearest neighbor algorithm, artificial intelligence algorithms, which are very broad range of different algorithms like deep neural network. And the results, and once you get results, usually it means that you have the prediction of biological activity and design of different molecules. So what exactly is the chemical space we are trying to analyze? The chemical space is the space which represents all the molecules which we probably could think of. It's the huge number of molecules. Usually it's a kind of you can imagine it as a universe, and comprises the huge number of molecules, such as from 10 to in the 6th power to 10 to the 9th power molecules. Sometimes people even increasing this to a 10th in the 14th power. And from this realm of the molecules, we would like to find some molecule which is probably very important molecules in terms of affinity, bioavailability, pharmacokinetics, pharmacodynamics, binding, degradability, druggability, and some other parameters. And the idea is to find the needle in the haystack, and sort of you have a haystack, and this is the needle. It's really hard to find this needle. And the chemoinformatics paradigm is actually to find this needle. So you would take this chemical database, trying to screen it or sieve it through this funnel, and as a result, you will come up with a small number of molecules. So actually, the chemoinformatics is all about the filtering of different molecules and filtering the chemical space. And by which means it's possible. It's possible when you have different methodologies in terms of the filters, similarity search, Kursor, structure activity, structure properties, Farmakophore, Dokken, and so on. And then you got most of the molecules, they will come up as inactives, and the small portion of molecules, they will come up as actives. Okay, what else? What else is the chemoinformatics learns from the experimental data. And sometimes experimental data learns from chemoinformatics, like the model learns from the experimental data, or the experimental data learns from models. This is the paradigm which has both ways. And you can represent these paradigms using some kind of a triangle, where the top is the knowledge and the bottom is the data, and they interact with each other by means of the information. And when you're going down, it means you have a deductive learning, and when you're going up, it's some kind of the inductive model. So these strategies are widely used in the chemoinformatics, actually in the science too. And as exactly what we have here, we have some kind of a data, and we will try to understand how this data interacts with the protein, and we will get some knowledge of that. So by clustering, we will try to reconstitute actives and inactives, and by the Gibbs free energy of binding, it's possible to describe the protein ligand interaction. Okay, so why robotics? Because robotics is a kind of this is the experimental tool. Sometimes people are using robotics to describe the computational data, like well-known, probably the next the Nobel Prize in a protein folding like AlphaFold. It's also kind of a robotics, as you see. It's some kind of a machine learning algorithm. But of course, in a narrow way, it's in a strict way, the robotics is something which you do some which you will perform in the lab. So it's these are the wet experiments. And why we need these wet experiments for chemoinformatics? For the obvious reason, because we have a lot of testing samples here, like in our publications, because so far they are just hypothetical entities, starting from Alzheimer's disease to some zinc databases where we screen your natural substances. Here we have, as I understand, probably a couple of, probably hundreds of substances they were screening, and we will probably trying to come up with tens of the molecules. Of course, it's better to use robotics here, because if you will perform something like in a consecutive way, it will take a lot of time. So obviously, robotic will enhance and increase the productivity and high throughput screening efficiencies when combined altogether. And this is also this paper from my very old publications. When we are trying to implement lead expansion and a virtual screening for the HIV proteases. This is extremely important for some diseases, so far incurable diseases. And if we will combine these high performance robotic facilities, we will of course have more chances to treat incurable diseases such as HIV or cancer. The other thing is what is exactly important in terms of the blood-brain barrier. There were some indications on the blood-brain barriers already, and the blood-brain barrier is something which is important in the treatment of neurodegenerative diseases, because the substance should permeate through the brain. When you will inject the substance into the bloodstream, it will go to the bloodstream, and then there is a formidable task as known as a blood-brain barrier, which will protect the brain from the different substances, either from the internal or the external, like symbiotics. And this is the rough scheme. We'll show, we'll give you a hint why it's a real problem, because only 2% of drugs is able to permeate through the blood-brain barrier, and of course 19% is not permeating. So the majority of drugs should be either enhanced in terms of the blood-brain barrier permeation, or you might want to think of some kind of excipients to surpass this problem, like calixarenes or cyclodextrins or calixarenes, like some kind of a formulation substances, if you will try to formulate. So you might probably increase the blood-brain barrier permeation as well. And what kind of substances we were already checking in our experiments, which are probably have some properties in terms of the blood-brain barrier permeation. The CNS active substances, some carbon nanoparticles made of, they're actually hybrids. So we have a carbon nanotube with some kind of a linker, and then this linker is connecting this nanotube with the drug. And excipient substances like cyclodextrins. We are using different techniques like in silico, in vitro, in vivo, sometimes even ex vivo, to investigate this permeation. Okay, so a couple of words on what we are doing here in the Department of Infochemistry in the Laboratory of Chemoinformatics in terms of the drug design. So this is a kind of a scheme which we developed. We are using the scaffold of interest, and by applying some rules like organic synthesis rules, like quick reaction, we were able to synthesize various molecules which are able to permeate the blood-brain barrier. So far, we were able to synthesize like 20,000 different substances using some descriptors like SASA, like molecular volume eccentricity, and the other descriptors comprising like 13 different descriptors. And on the other hand, we are also doing some research to understand the interaction of aptamers. The aptamers, they are substances made of the DNA molecules, and they are particularly important to bind various molecules like toxins, antibodies, various proteins, probably some targets, some transporters. And this is a scheme and how we are trying to model this process, starting from the aptamer sequences to the 3D structure, and then to the interaction of the aptamer with some targeting molecules. It's still in the progress, so I'm not going to give you a lot of details on this scheme. Okay, and this is the information where it was already published. This is the study. This was already published, where we're trying to synthesize the molecules, like some kind of virtual database, which was made from the click reaction, and substances, as you can see here, they are quite promising to permeate through the blood-brain barrier. And after that, we were able to link these particular substances with some receptors in the brain, since they're permeating like dopamine receptors, and we found that these substances are promising to inhibit these dopamine receptors to treat some diseases like the depression, for instance. Here is again from our previous study, and how the ligands are interacted with the choline transporter, and this choline transporter is expressed in the BBB and can be used as a kind of enhancer or a protein which probably pump the molecules up to the brain, will help the ligands to overcome the restriction of the blood-brain barrier. Okay, so what is next? Can be done in terms of the high throughput screening, virtual screening of the different drug-like molecules. This is the combination of the algorithms, and to create it, in order to create some kind of software for the various purposes, for for the researchers from different fields, like a biomedical field, because the people from the biomedical, they're not really very enthusiastic using some scripting, so they would prefer the point and click software, and which is exactly what we were trying to reach here. So we integrated the algorithm, and the algorithm is called is NeuroClick. And as you can see, this algorithm is just a very simple thing, how to create the synthetic database using some descriptors, some standard descriptors which are known as the Lipinski rules. We also integrated some blood-brain barrier models like Clark model and Richter models. You can actually scale up different models using different thresholds, using different descriptors. So it's quite manageable to change different parameters. And as an output, you have different substances coming out of the click reaction, which have different number of different number of molecules, the data in a database, different number of hydrogen atoms, acceptors, atoms, molecular weights, also different log P in terms of the hydrophilicity, hydrophobicity, and of course, the most important thing, they can be analyzed as a log BB, which is the main parameter to predict the permeation through the blood-brain barrier. Okay, and so this is just a couple of words. This is not really the published data. I'm trying to investigate the interaction of different proteases. These are HIV proteases with different ligands, and here is to understand how different proteases from different viral phyla are related in terms of the dimerization distances. So if you will imagine the protease, the HIV protease, it has two dimers, and these two dimers are actually separated at some distances from each other. So if we would try to analyze the interaction distances between the interaction force between these two dimers, you can see that you have some correlation in terms of the interaction. So the HIV, for instance, the human HIV, they are having less interaction force, and some feline HIV and some kind of monkey HIV viruses, some kind of monkey immunodeficiency viruses. They have the highest force, which is remarkable. And by using this information, one could also create some kind of algorithm. And this algorithm is particularly important to remove the bias of molecular docking studies, because I mean, this is the well-known fact. If you will try to investigate the protein-ligand interaction, you will come up with some differences in the interaction because of stochasticity, the algorithm stochasticity, and the imperfection of the genetic algorithm widely used in the docking experiments. As you can see here, for instance, if you will try to do the experiment 3 times or 5 times, you will have a huge deviation of results based on the stochasticity of the algorithm. And by using this information, by using the information of the dimerization and the interaction force between different monomers in the HIV protease, one could create some kind of algorithm to calculate the Gibbs free energy of binding, which corresponds to IC50, as you can see here, described by the S curve, and the delta G is called here as a transition delta G, which has no deviation imposed by the single experiments. And from this experiment, one could precisely say what is the exact interaction protein ligand interaction between different ligands in a database. This information is also a very good correlation with the phylogeny data. As you can see, we also have some kind of clustering here. Then the group of HIV viruses is separated from the group of the other viruses, like the simian immunodeficiency virus or feline immunodeficiency virus, or the viruses representing the different phyla. Okay, so what's next? Why the robotics is important for the proteomics, for instance, for the structural biology and the proteomics. And here, again, we are doing something which is very important to investigate the main document biology, like the protein folding. And the protein folding is actually the kind of a structural representation of the protein function. As you can see here in our current publication, which dealing is with topological bioscaling analysis is a universal measure of protein folding. And in this paper, we were trying to understand how unfolded structure will go through different transition states and will reach the complete folding. So by using some mathematical function, we devised this process and calculated some alpha, some kind of alpha constant, which is giving us the dimensionality of this process, of starting from one dimensionality to the 3D dimensionality of this process with a high correlation. We also checking different parameters like what is the ideal, for instance, ideal deviation, the deviation from the ideal structure, and the ideal, I mean, the ideal structure in terms of the X-ray crystallography, because here we also rely on the experimental structure. So we are trying to investigate how one structure which is folded and the other structure is not folded are related using some algorithms like Monte Carlo algorithms. And again, we have found some clustering here where we have the very high score when the protein is unfolded and very low score when the protein is folded. So the robotics probably is important when you have different protein conformations, and if you want to compare these conformations, like usually the Monte Carlo algorithms here are able to generate one dot is actually one conformation, and if you want to confirm it experimentally, you have to reproduce this in the lab. So for this point, the robotics technology might reproduce this kind of big data coming from Monte Carlo algorithm as well. Okay, so I'm not going into detail, just a couple of words on our research project. We are trying to investigate the mutational rate of the mutation, the SNPs, single nucleotide polymorphisms within some proteins, which are called defensins. They are very important for innate immunity because they are responsible for different processes in the immunity, protecting the human body against different pathogenic factors. They are important for hemotaxis, wound repair, mitogenesis, abscinization, and so degradation of mast cells, degranulation of mast cells, and so on. So they're extremely important. They're also important to protect us from bacteria because they are interacting with the bacterial cell wall with some lipids in the cell wall, and by doing this, they will undergo the bacterial death. And by using some computational algorithm, we were trying to investigate what are the mutations, what are the SNPs, what are non-synonymous mutations within these proteins, and how these mutations are actually important to interact, to make this protein more susceptible or less susceptible with a higher or low affinity to interact with the lipids of the bacterial cell wall. Actually, this was the hypothesis, and we did some bioinformatics magic here, and we found some SNPs like SNPs which are given very unstable impact on the protein, like in the position of 62. So you can see here that if you will mutate the protein, it will actually decrease the stability of the protein and probably the function too. And we would try, the next step would be to trying to correlate and how the function of protein is also impaired by the mutations. And we found that some mutations are taking place at the at the interface where the interaction with protein ligand interaction is occurred. And we analyze this phenomenon found in indeed, if you are mutating it, so you are probably decreasing the protein function, and the protein ligand interaction will go down. Also, a couple of words on robotics. Here we have a project. I have collaborations as I'm a visiting professor in the University of Würzburg, Department of Bioinformatics, the Laboratory of Synthetic Biology. And here we have some synthetic biology experiments. The idea is to mutate the enhanced GFP to investigate how salt bridges are actually important in a protein function. Probably we will enhance or we will decrease the fluorescence of this protein. On the other hand, it is not very well known how this salt bridges are important in the folding of this protein. And by doing this, we will implement site-directed mutagenesis, as you can see here, using bacteria express some vectors and which are expressed in the bacteria. So we created a lot of mutants, and these mutants were compared with the reference. And the next step is we were trying to investigate what is going on with the structure of the protein using this mutation. And by doing this, we were able to create some very sophisticated in silico models and compare with experimental data. So as you can see, we have a lot of robotics. It takes extremely long time so far, more than a year, I would say, up to 2 years. So again, if we would use the robotics, we would come up to we will squeeze this time to a couple of months or so. So this would be extremely helpful in our case. Okay, this is again some kind of a robotics facility I would like to show here. We are starting from high performance computing down the hill to the experiment. We have some kind of in vitro robotic facility, and from this robotic facility we will come up to the heat molecule. Of course, we are relying on the large libraries of the molecules and sometimes the libraries of target proteins we would like to analyze. Some laboratories are already implemented for the research in Germany. There is a Fraunhofer Institute in Germany. I was a part of this institute previously when we were together writing a lot of grant proposals using some kind of tissue engineering facility. And as you can see here, it's a robotics facility in Würzburg where they're implementing some kind of biovask models to investigate how actually molecules they're permeating through the blood-brain barrier, but they are not using the classical transfer models. They're using some kind of tissues coming from the intestines, and this intestines is used as a so to say the bloodstream, and some kind of a dynamic model. So you can see it in dynamics. If you will try to implement something which is based on a regular basis in our lab, in Professor Foster's lab, and I was doing a lot of experiment myself, it's not really a dynamics, it's a statics, and a lot of parameters are missed. So by using this very sophisticated robotics, we were able to also monitor the permeation by mimicking the actual bloodstream. Okay, the other robotic facility is probably important to use in the evaluation of the toxicity of the substances. Here is the platform for the toxicity, for the in vitro toxicity. You can order it, and toxicity platform is very useful when you would like to apply like toxicity tests, like the MTT test, when you have a MTT conversion to a purple pharmazone, and by changing the color, you could say whether the molecules are toxic or not. And this also relates to our previous publication, where we're trying to investigate the potential toxicity of unmodified and modified cyclodextrins using the blood-brain barrier and the T-level cells. The only difference is, with that time, we didn't really use the robotics toxicity platform. And to do these experiments, we tested only up to 3 to 5 different drug-like molecules modified complex with the cyclodextrins. Okay, so this is our robotic facility here in the ITMO. It's called the QCA robotic arm, and you can program it to do the chemical experiments. Hopefully, I'm looking forward to implement my necessities in chemoinformatics and bioinformatics to expedite the experimental settings, and to produce the data, and to squeeze the time and enhance the chemoinformatics platforms, not using the chemoinformatics, very sophisticated chemoinformatics algorithm, but also very sophisticated robotics settings. Okay, this is all about it. This is all what I wanted to say and to highlight. And thank you for your attention. And